Hello everyone, I am Mrs. Ekta God with my new one video for the topic plant physiology. In this video, I am going to take the plant growth and development. Please do comment if you like and you are getting benefit from my videos. I will be very much thankful to all of you. What do you understand by development? So, development is the sum of two processes. Development means growth and differentiation. Growth is irreversible, permanent increase in size of an organ or its parts or even of an individual cell. Growth is accompanied by metabolic processes, both catabolic and anabolic, that occur at the expense of energy. That means both catabolic and anabolic process use the energy to provide the growth to the living creatures. Plants show open form of growth. That means new cells are always being added to the plant body. Which part of the plant provide the primary growth? So, apical meristem contribute primary growth to the plant. Another one is the lateral meristem, which contribute lateral growth and causes increase in girth. Vascular cambium and core cambium are the components of lateral meristem. Growth at cellular level is principally a consequence of increase in the amount of protoplasm. Growth is measured by a variety of parameters like increase in fresh weight, dry weight, lengths, areas, volume, and cell number. Period of growth is generally divided into three phases. Number 1, meristematic phase. Number 2, elongation phase. Number 3, maturation phase. Cells in meristematic phase are rich in protoplasm, possess large conspicuous nuclei. Cell walls of meristematic phase are primary. Phase of elongation characterized by increased vacuolation, cell enlargement and new cell wall deposition. Cell of maturation phase attain their maximal size in terms of wall thickening and protoplasmic modification. The increased growth per unit time is termed as growth rate. The growth rate shows an increase that may be arithmetic or geometrical. In arithmetic growth, only one daughter cell continues to divide while other differentiate and matures. Root elongation at constant rate is the expression of arithmetic growth. It proves linear growth and can be expressed as LT is equal to L0 plus RT. It represents arithmetic growth. LT is the quantity or length at time t. L0 is the initial quantity or length at time 0. R is the constant rate of increase or elongation per unit time. T is the time. This formula describes a situation where a quantity increases by a constant amount over equal intervals of time. A classic example is the elongation of a root at a constant rate, where plotting the length against time results in a linear curve. LT is equal to L0 plus RT is the mathematical expression for arithmetic growth. R denotes this equation. So, growth occurs at a steady pace and rises in an arithmetic way. Between the two resultant progeny cells, only one cell is permitted to divide. In geometrical growth, both daughter cells continuously divide. It attains sigmoid curve if space and food is limited. For example, trees with seasonal activities show sigmoid curve. Exponential growth or log phase of sigmoid curve can be expressed as W1 is equal to W0 e raised to the power RT. Here R is referred as efficiency index. Here W1 is the final size or quantity after a certain time. W0 is the initial size or quantity. Small e is the base of natural logarithm. Small r is the growth rate, which is a constant factor indicating how quickly the quantity is increasing. T is the time that has elapsed. Quantitative comparisons between the growth of living system can also be made in two ways. Number one is absolute growth rate. Number two is relative growth rate. What do you understand by absolute growth rate? It is the measurement and comparison of total growth per unit's time. And what do you understand by relative growth rate? It is the per unit initial parameter. That means increase growth divided by the initial parameter and multiplied by 100. Water, oxygen and nutrients are essential for growth. Plant growth and further development is intimately linked to the water status of plant. Water helps in turgidity, enzyme activation. Oxygen helps in releasing metabolic energy. Nutrients helps in synthesis of protoplasm and act as source of energy. Optimum temperature range and environmental signals such as light and gravity also affect growth. At the time of growth, undifferentiated cells are shoot and root apical meristem. Differentiated cells, those have lost the division ability, are parenchyma, collenchyma, sclerenchyma, primary xylem and phloem. Dedifferentiated cells are those regain the ability to divide. These are interfascicular cambium and cork cambium. Redifferentiated cells are those can lose the ability of division permanently. They are secondary xylem and phloem, cork and secondary cortex. If plant growth is open, it can be determinate plant organs. If plant growth is intermediate, then it can determine shoot and root apices. Plant differentiation is also open if same meristem have different structures at maturity. Final structure at maturity of cell or tissues is also determined by location of cell within. Development includes all changes from seed germination to senescence. Plants follow different pathway in response to environment or phases of life to form different kinds of structure. This ability called plasticity. Heterophyly in buttercup, cotton, coriander and larkspur is an example of plasticity. Development in plants is under control of extrinsic conditions like light, temperature, water, oxygen and nutrition. Development in plants is under control of intrinsic conditions also. Intracellular conditions are genetic and intercellular conditions are affected by the PGR factor. Plant growth regulators are small, simple molecules of diverse chemical composition. Plant growth regulators are auxins, cytokinins, gibberellins, abscisic acid, and ethylene. Auxins are indole compounds. Cytokinins are adenine derivatives. Gibberellins are terpenes. Abscisic acid are carotenoids. Ethylene are the kind of gases. What do you understand by PGR factor? PGR is the growth promoting activities. Growth promoting activities are cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. Plant growth promoters are auxin, gibberellin, and cytokinin. growth inhibiting activities of PGR, dormancy, abscission, response to wound and stress. Plant growth inhibitor is abscisic acid. Ethylene is both promoter and inhibitor, but it is largely an inhibitor. So it was about the plant growth and development. In my next video, I will take the revision of plant growth regulators one by one. So be with me. Have a nice day. Have a happy day. Bye bye.